Not sure if Duncan and Isabella told you that right after spring break, we're flipping what's on the syllabus in the order of lab experiments. So Tuesday we'll talk to you about applied fermentation options, and then Thursday we'll do the fungi stuff. Okay. Did any of you take plates to go capture fungi? Always revealing what you get. I'm gonna do that too. Yeah. So we'll see what we end up with. Um, okay, so today we are starting a new topic on I should probably rename it. It's put down as gene expression, I guess, regulation of gene expression on the syllabus, but really what we're talking about is microbial communication. How do microbes communicate and coordinate their behavior? And so this started off with the idea of quorum sensing. And the pioneers in that field of research, Dr. Bonnie Bassler and Peter Greenberg, were the early pioneers on this idea. And I'm going to post a TED Talk in the readings and videos section of EDUCAT. It's an older one, but it's still really good on how bacteria talk to each other. And so Dr. Bassler is one of the pioneers, and like she does all kinds of cutting edge research. She also focuses on Vibrio cholerae that causes cholera. And they do all kinds of interesting quorum sensing in order to make people sick. So I'll post that. And then, so this idea of how do microbes assess their environment in terms of who else is around them. You know, they're constantly assessing their environment for what kinds of nutrients, carbon sources, energy, electron sources, all of that. But they are also very social. So there's a whole field of microbiology called sociomicrobiology, believe it or not. And they use some of the same concepts as what you would think about with sociology and human interactions. And so it's interesting to read some of those papers. But when this was first being proposed, like, do bacteria really communicate? A lot of people were like, yeah, probably not. You know, they're not that sophisticated. But Bassler's and Greenberg's work was quickly showing that they do interact with each other and communicate. Obviously, they don't have ears and mouths like we do, so they don't use oral communication, but they use chemical communication for sure. And so some of the terms that have been given to these chemical signals are called autoinducers, pheromones. The pheromone term is taken from, you know, the idea that animals have pheromones as a means of chemical communication. So you'll see those in the literature quite a bit. So you might hear me saying those. The figure here is trying to show that you have a microbe on the far left that is busy making some chemical communication signals, and then they excrete them out. So what, in general, what they think happens is the bacteria are pretty steadily doing this. So it's kind of like a constitutive thing. They kind of steadily make low levels of this chemical, all right? And excrete it out. And then the recognition idea, we're going to talk about how do microbes pick up on this signal. And there's different strategies. I'm only going to give you some basic ideas because some of the strategies are very, very complex and sophisticated. So we'll just do simpler versions, but that middle picture is trying to show that you have to have that signal dock into some kind of receptor or somehow have this recognition in some manner. And then you see this last shift. In this case, they're showing it could trigger the bacteria to shift its gene expression to maybe become very virulent and pathogenic, maybe to glow, and some other behaviors. All right, so this was actually first discovered in bioluminescent bacteria. So the bacteria that hang out with squid, cute little squid, and the if you looked at this idea at all, these squid have these organs right below their eye that allow them to glow in those regions. There's also fish that do this. And so the idea that bioluminescence might be under some kind of sophisticated control was not too surprising. They thought, though, that this quorum sensing idea maybe it's just limited to bioluminescence because it is a pretty unusual trait, you know. And so they thought, well, maybe this is all just to do with the bioluminescence idea. But they did look at this more carefully in other non-bioluminescent bacteria, and they found there's all kinds of bacteria that are doing this. It's not just the funky ones that do the bioluminescence. So Vibrio fischeri has been renamed to Allevibrio fischeri just recently, but you'll see tons of papers published where they just call it Vibrio fischeri. And what they discovered is that gram-positive, so Vibrio fischeri is gram-negative. And so they studied the gram-negative way of doing communication first, and then they looked at gram-positive. They said, do gram-positive bacteria have this, and do they do it the same way? And back then with that research, they do it different. And so I'm going to show you essentially two different strategies that have been well studied on how they do this kind of communication. Whoops. And so there's the cute little squid, the bobtail squid. And then figure C and D are the bioluminescent bacteria. So just under normal lights in the lab, if I gave you a plate, it would look like figure C. It wouldn't look like anything extraordinary. But then if I told you to go into the walk-in warm room in the lab and close the door and make sure all the lights are off in there where it's pitch black, then you would see them bioluminesce with just the naked eye. You don't need any fancy like UV light or anything like that. Just with the naked eye, you let your eye adjust and you can see them glow right on the plate. So glowing bacteria was very, very interesting to a lot of scientists. They were trying to figure that all out. And here's more plates showing what they can look like. So remember, each colony on the plate is millions of Vibrio fischeri. So you have lots and lots of these cells all in close proximity, chemically communicating, and then they coordinate their behavior to go ahead and bioluminesce. And so the question remains, why are they doing this? And we'll actually talk a little bit more about the ecology of this later. And also Dr. Bassler's video, her TED Talk video, she gets into that. Like, why would they have this kind of behavior? Why would they associate with squid and other marine organisms? What could it give? What's the win-win situation here? So she has some nice ideas on that. But first, let's go into like the basics of how are they actually doing this. And so with gram-negative, we know they differ from the gram-positive with their cell wall type, but they also seem to have this ability to produce these chemicals called N-acylhomoserine lactones, which are abbreviated HSLs, or sometimes they abbreviated AHL, same thing. So the lactone ring is on the left here, the brown, that's the lactone ring. So homoserine lactone. And then the acyl side group is the string of carbon and nitrogen there. So the acyl side group. And then, they have it right there, nicely labeled. So here's the lactone ring, homoserine, and then the acyl side group. So when they were looking at Vibrio fischeri and how this is working, what they realized is when they worked with it in the lab, they could detect that Vibrio was making this chemical at low levels pretty steadily. And so it's a constitutive process. And so it's their way of sending out a signal, and they're kind of waiting to hear back. Like, is anyone home? Is anyone else out there? And so what happens is they make the signal, they excrete it out, and they're doing it, each cell is doing it just at low levels. But then if you can imagine 1,000 of these cells all hanging out with each other, and they're all doing this, then that chemical gets to a higher concentration around the cells. And so we say that the signal accumulates at high densities. So once there's enough cells, enough cell density, high enough population, they're all excreting this chemical, you get a pretty decent amount of that chemical. And once it crosses a threshold, so to speak, it'll start to diffuse back into the cell. All the cells in that area, that chemical will start to diffuse back in, and it binds to a regulatory protein inside the cells. And then that regulatory protein, now that it has that chemical signal, it will go to certain aspects of the chromosome and alter gene expression. So this is kind of like the cellular molecular way that this is happening. And basically, it allows the bacteria to be assessing how many of, so with Vibrio and them hanging out with the bobtail squid, they're constantly like, how many other Vibrio are around me? Who's there? How many? How many? And they are able to sense how many are around, especially as we trigger that threshold level and that chemical starts to diffuse back in. And then that allows them to shift their behavior into a group behavior. Let's do something as a group that's more effective than if we tried to do it all by ourselves. And so bioluminescence fits that bill. If you are one cell and you're trying to bioluminesce, you're not going to glow very brightly, right? It's just, you're not going to be noticed. If there's 10,000 of you all hanging out together in the bobtail squid tissue and you all bioluminesce, then you're noticeable. And so this idea of they're shifting their gene expression in order to coordinate with each other and do a group behavior that is successful when there's a group of you, but not very successful when there's not a group of you, right? And so the bioluminescence idea is a very, obvious response that the bacteria are doing because they're glowing and it's very in your face. Literally, you can see it. So this is showing a section of the chromosome and different genes on the chromosome. And this is for bioluminescence. And what they're showing is that some of these genes are turned on just constitutively, small amounts of these chemicals are getting made. And they're trying to show that the LuxI gene, you don't need to know the name of the gene, but the LuxI gene is, once it gets turned on and transcribed, you get the homoserine lactone that gets excreted. And then if lots and lots of the cells are all doing this, it will bind to, the chemical signal will diffuse back in and bind to the section of the genes that cause bioluminescence, right? In this particular case. So luciferase is what we see when we look at those colonies bioluminescent. And so the chemical docks to the LuxR, this is the regulatory protein, it'll connect onto that, and then it turns on, that will allow it to turn on the genes for glowing. Otherwise, they won't glow unless there's enough of them around to make it worthwhile and make it beneficial. So we say it's at high cell densities that this behavior, this bioluminescence occurs. And that's what they're trying to show here, that you have to get enough of the biomass, enough cells, all close to each other doing this chemical signaling, and then that threshold gets triggered, the signal comes back in, and you start to see this real big upswing in bioluminescence. So they say it's cell density dependent. You have to get a certain level or a certain population reached before you will have enough of the chemical signal flowing back in and binding to the regulatory protein and shifting gene expression. Here's another visual of this idea, again, with the chromosome, the stretch of genes involved. And so this one's a little bit nicer in that it's actually showing, they're just using a red, the lactone ring to show that this is a homoserine lactone getting excreted at low cell densities. All of them are just doing a little bit of it, but if you have 10,000 cells all around each other, then that signal starts to really accumulate and it'll diffuse back in. And now it's binding to the regulatory protein. And then that complex, that protein plus the chemical signal bound to it, will go and alter gene expression. In this case, let's go ahead and bioluminesce. questions on that basic process? There's a video that I posted that you can watch when you're studying that's in the slide set there, so you can use that. So they discovered it in Vibrio. They're like, well, that's really cool. It allows the cells to only bioluminesce when there's lots of these cells all around each other. And then they figured out what the homoserine lactone is. And then they started looking at related organisms to Vibrio, unrelated organisms, and they're like, do all bacteria do this, or is it just unique to Vibrio? So here you can see that there's a range of different microbes, and they're not all Vibrio. And what they're showing here is the structure of the homoserine lactone, and then they're saying this R section can vary. Okay, so sometimes it's just a methyl group, which Pseudomonas does that, and then sometimes you have longer chains here. So that R group, the side chain can vary in its length. But pretty much the rest of it stays the same. And so you can see that these are all gram negative. Yep, all gram negative. And they're all producing a homoserine lactone, but it can vary a little bit. And so that's kind of interesting that that's kind of a universal basic chemical, and then they just play with the acyl side chain. And so that has some interesting implications too that we'll talk about in a little while. Here's another image of some of the homoserine lactones that have been isolated. So you can see as you just pan through this image that it's fundamentally the same. It's just the side chain that's varying in its length. And so, the question is, can some of these that differ in their length still be recognized by the same receptor or the regulatory protein, I should say? Or are these all unique and they can't cross communicate? So there's a lot of, initially there's a lot of interest in that idea, which we'll talk about. So here's another visual trying to get across the idea that if you have a group of cells all close together, all excreting a small amount of the homoserine lactone, eventually you'll trigger the threshold level and it'll diffuse back in, and there's your regulatory protein that will go. Now the chemical binds to it, and that will be the signal to the protein to go to the DNA and alter gene expression. So you have been impacted by this. If you've been sick by a bacteria pathogen, this has been going on in your body. I hope it's not happening currently in your body. I hope you're feeling healthy enough to go and enjoy spring break. But if you've had strep throat or something like that, you've had quorum sensing going on in your body and it was not working for you. It was working against you. And so it's often useful to think about like warfare or pathogen host interactions as you try to think about this idea of quorum sensing. Because if Pseudomonas gets in you, by the way, Pseudomonas aeruginosa is an amazing bacteria. It can be pathogenic to humans and to other animals and to plants. So it can cross over into the plant world and cause devastating crop failures there as well. But it can also be beneficial. So to certain plants, I know more about how it interacts with plants and with humans, but at least in the plant world, it can be beneficial in certain conditions, but it can also be deadly. So anyways, if you think about it kind of like warfare. So if Pseudomonas gets in you, and I talked about that case study where that guy went partially blind in his eye because he had Pseudomonas from the hot tub on his fingers and he scratched his eye. So what was happening is the Pseudomonas got into his eye tissue and it started excreting some homoserine lactones just at low levels. And then as Pseudomonas is hanging out there and dividing by binary fission and the population levels are going up, then all of those cells all excreting that chemical in small bits. Eventually there's like a trigger, threshold's been met, signal comes back in. And then what they do is they start to coordinate their behavior, shift their gene expression to attack the host. So if you are an army, if you're in warfare, and you have a unit of 50 soldiers. And they're up against a sophisticated enemy. You know, the immune system is incredibly sophisticated. You might want to wait and wait for your other units to come join you before you attack. And so if you think along those lines, I think it can make a little bit more sense of why they're doing this and what's the result. And so 50 soldiers versus, 5,000. It'd be better to attack when there's 5,000 of you. Then you have a chance. And so that's what we mean by, this group behavior that's beneficial. They can't really do that behavior when there's too few of them. It doesn't really help them in any way. But if there's a lot of them doing it all at the same time, then they can coordinate and hopefully be successful. Now, from Pseudomonas' perspective, when it got into that guy's eye, they weren't trying to kill the guy necessarily. They were just after the nutrients. And so they were trying to excrete out toxins and virulence factors that would allow for cells to break open so they could get more nutrients and all of that. Unfortunately, it caused that guy to go temporarily blind until he went and got antibiotics to clear. So, from the pathogenic perspective, if you've been sick with any kind of bacteria, that's been happening in your body. And it is, initially you don't know you're sick at that stage when they're quorum sensing with each other. You don't know you're sick with the pathogen because they haven't revealed themselves yet, because they haven't attacked you yet. They're trying to stay under the radar. So this is a silly visual, trying to get this idea across that they need to be able to assess how many of Pseudomonas are around me, how many of me are around me, because I don't want to attack if there's too few of us. It will probably wipe us out, especially when you think about how sophisticated animal immune systems are. It wouldn't take long if there's not too many of them to get wiped out. So again, this is just trying to show that it's linked to the population of the bacteria. And once you get a high enough, so it's density dependent, once you get a high enough amount of cells and a chemical signal, then you'll get a shift in gene expression because of that quorum sensing going on. So pathogens, all the pathogens so far that have been studied have shown some ability to do this. Yeah. And so when I'm hunting for quorum sensing signals, I tend to, because I want to get a fair dose of it, good concentration of it, I tend to grow the bacteria until they're at late stationary and late exponential and starting into stationary. That's usually when I can see maximum quorum sensing signal. Okay, so this was all with gram-negative bacteria. And so they started to hunt with this, like how many bacteria can do this? And so many that were studied, it's like, yep, they're doing it. And so then they said, well, what about gram-positives? And then they realized, okay, gram-positives do it a little differently. Okay, so this, it's a little bit more complicated because some of the basics are the same. So, except that they don't have homoserine lactones. They have something that they use instead called peptides, which are just small strings of amino acids, okay? So they use amino acids strung together in certain combinations. And the initial term was called peptide pheromones because it just sounded catchy. And the pheromone idea, people could generally understand what a pheromone was. And so it kind of fit of what it's doing for the bacteria. So again, it's self-produced at low levels pretty steadily as the bacteria is just hanging out. It gets excreted out into the environment. It accumulates at high cell densities. And then again, it will alter gene expression. They used to say it will turn on gene expression, but the better way to phrase it is alter, because they learned not too late into this that it can turn on gene expression, but it can also turn off gene expression. So just say alter and you're safe. It alters gene expression in some way. So these are some examples of the peptide pheromones. So all those letters, those are different amino acids that are strung together. And you can see Staph aureus, which you've worked with. So there's different strains of Staph aureus here, and they have slightly different peptide pheromones. Then they started looking at others. A lot of what they looked at initially were human-associated microbes, because of course we were very curious about that. And so Bacillus subtilis, Streptococcus pneumoniae, obviously can cause pneumonia. Staph aureus is super common on your skin. Lactic acid bacteria in the food industry. They were very interested in this because if you make yogurt or anything like that with lactic acid bacteria, being able to understand how to optimize their growth is a good idea. And then the Streptomyces species were also being studied. They're kind of an odd duck because they don't seem to make peptide pheromones, even though they're gram-positive. So I'm going to kind of leave them and talk to them about them separately. They're weird. But basically, it's this idea that they're still making the signal, and then they excrete it out, and then it accumulates at high density, and then somehow it's altering gene expression. But it turns out it wasn't the same way as what we saw with the gram-negatives. So they use a more sophisticated system called a two-component signal transduction system, which is not, this isn't unique to bacteria. Like plants do this, animals do this. So this is a way to take some external signal and transduce it into the cell to shift gene expression. Okay, so transduction, signal transduction is just saying you have to take that signal from the outside of the cell and somehow tell the inside of the cell what to do with it. And so it's a signal transduction process or system. And so two-component means there's two main factors involved. There's a sensor kinase, which is just a protein embedded in the cell wall, cell membrane, that's assessing and looking for that signal, so to speak. Okay, so it's waiting for a signal. And then inside the cell is a response regulator. So those are the two components. Okay, two players. Okay, so you have to imagine these gram-positives, they're excreting these peptide pheromones out, and then what happens is you get, so the pink balls are the peptide pheromones. Here's your sensor kinase, so you see it's docking into the sensor kinase. And what that does is it triggers the kinase to get phosphorylated. So there's ATP being used, so it's energy cost. The ATP donates a phosphate over to the sensor kinase. And then I like to think of the sensor kinase and the response regulator kind of like relay races, you know, when you have the baton. And so I think of the baton as the phosphate. And so the sensor kinase is like, okay, I have the phosphate and now I have to hand it off to the response regulator, and then the response regulator takes off and does its lap. And basically what happens is once the response regulator gets that phosphate attached to it, it kind of wakes it up and goes, oh, and now it's my turn to do my job, which is to go to the DNA and shift or alter gene expression. Hopefully in such a way that allows for a group behavior so that they can accomplish something now, because there's enough of them, they can accomplish something successful as a group. So that's the two component. You have the sensor kinase, response regulator, and then the signal transduction is the idea that you take this peptide pheromone signal and you transduce it into the cell and alter gene expression. So this is a universal mechanism. Like I said, bacteria came up with it first. They were the first on the planet to do this, but it's been co-opted and even more sophisticated layers of signal transduction in some of the other organisms on the planet. So this is not unique, but it's a pretty straightforward system in order to be able to make sense of the signals and shift the behavior of the organism appropriately. Questions on the two-component signal transduction? Here's another image of it on paper. So I guess the only downside to this really is there is a little expenditure of ATP. You have to invest a little bit. So when they say cell density dependent phenotype, remember phenotype is the outward expression of the genes being, turned on or off. And so that's kind of like when I say the behavior, that's what I'm referring to. It's the phenotype of the organism. So you've already seen this idea when we were looking at how certain bacteria pick up naked DNA in the environment when we're looking at transformation. And we talked about how with gram positives, they tend to build these transformosome complexes so that they can bring in a little bit of the DNA. And that's actually quorum sensing related. It's due to quorum sensing that allows them to do this. And so all of these CSPs that are floating around, those are the signals that are then docking here. So this is a type of sensor kinase, and then it gives the phosphate to the response regulator, and then that alters gene expression. So essentially, that idea is under control of quorum sensing. And so again, it kind of makes sense if you have a lot of cells around you, there's probably a certain percentage that are lysing and breaking open too. So there's probably a lot of naked DNA out there. So it kind of makes sense that they'll shift into this behavior when there's a lot of cells around them. Okay, here's one slide showing the comparison of gram negative versus gram positive. You can see the gram positive is a little bit more busy, a little bit more steps to it, but both very effective. So when they first looked into this, they're like, okay, gram positives do it this way, gram negatives do it that way. Well, the more you look in science, the more complicated it tends to get. And so they realized after more study, oh, one species can actually have multiple quorum sensing systems that it runs and operates. And so the work that Dr. Bassler is doing with Vibrio cholerae, it is pretty gnarly in terms of how many quorum sensing systems it can have up and running. And so I'm not going to get into that. What I want you to know is the basics. So I'm going to ask you, what does a gram positive do? What does a gram negative do? But we're not going to get beyond that too much because realize that in reality, they've discovered certain species that can kind of do both of these and even other things I'm not talking about. And so their ability to be pretty sophisticated is, I guess, it wasn't a surprise to the microbiologists, but it was to a lot of other non-microbiologists. So here they're just kind of doing this overlap of, all gram positives tend to have these peptide pheromones and gram negatives do these homoserine lactones, but then they've discovered a bunch of other things showing up. And then where these two overlap, you see this AI2 idea. So that is a weird chemical. Looks very different than a homoserine lactone or a peptide. And so both gram positives, early on, they discovered both gram positives and gram negatives can both produce AI2 type chemicals in their quorum sensing. So then the question is, okay, if they're both making it, can they both understand it then? If the gram positive makes it and excretes it out, can a gram negative nearby understand that? And the short answer is yes. And so that comes up with the idea that you can have different species hanging out with each other and they're all capable of kind of hearing each other talk and understanding the different languages. And so that has been another area of research. Okay. One last group here. The Streptomyces, I said they were odd. So they're gram positive. They're just odd bacteria to begin with in many ways. They're gram positive, but they look like petite fungi because they form mycelial growth forms. So they have long hair-like growth. They form spores in an interesting way. They're super common. Pretty much any continent that's been studied, they're all over the place. They love soil. And you have experience with them. I would wager all of us in this classroom have had experience with streptomyces if you've had to take antibiotics. They still grow streptomyces in labs to extract the antibiotics because the antibiotics are so complex. They don't know how to do it synthetically. So they just make the bacteria do it and then they purify it. Yeah, there's all kinds like streptomycin. You've ever heard of that one? You know, streptomyces, streptomycin. So they actually named it after streptomycin. But there's all kinds of antibiotics. The vast majority of antibiotics that are in play right now with humans and other animals are made by streptomyces. So that's one of the reasons why I got very interested in them, because I was interested in using them to help plants defend themselves against pathogens. And often the way to do that is to kill the pathogen through antibiotic use. And so they have to be different. They aren't like the normal gram positives in terms of what the signal is. They are using chemicals that look like homoserine lactones, even though they're not gram-negative bacteria. So I'll show you a picture of what I mean in a minute. But this is what they look like under the microscope. This is just the methylene stain. But they do stain gram-positive. They would look purple with crystal violet. But you can see the hyphal growth form. So they look like fungi. They're much more skinny. And the hyphae are very petite and skinny compared to fungal hyphae. But they actually thought they were fungi when they first discovered them. That's why the word the myces, that means fungus, but they're truly bacteria. And then they look like this on plates, agar plates. The colonies can be beautiful colors. I think I showed this to you already with the crystallized antibiotic that's oozing out. And so they are very interesting to look at, complex life cycle and prolific antibiotic producers. But this is what they do. So here, these are the two bottom images are true homoserine lactones from Vibrio fischeri and another gram-negative called Agrobacterium. And then all the rest are what different streptomyces make. And you can see the similarity. So the fancy word is they produce gamma butyrolactones. You don't need to know that, but that's the name of the chemical. These are gamma butyrolactones. And so they look very similar to homoserine lactones. So they got to be different in that regard too. All right, so here are a variety of gram-negative and gram-positive. Oh, there's the word gamma butyrolactone. The streptomyces. And then, yeah, so they're just showing you like the basic structure of the different chemical signals. And so this would be the phenotype or the behavior in this bar column. Like, what are they doing this for? You know, bioluminescence, conjugation. That's when they were doing exchange of plasmid strands. Exotoxins, so being pathogenic. Streptomyces like to produce antibiotics, and that's under quorum sensing. So lots of different phenotypes, essentially. But it does seem like the more they look, the more these different behaviors that are, you know, effective at high cell densities, when they look, it's quorum sensing that's controlling them. So we'll talk more about that and get a little bit more into the ecology, but I think I'm going to stop there. I did have a Google form question I wanted to ask you, but before we shift to that, are there any other questions on quorum sensing?
Notice that one of the slide was more academic. That we find something that we saw change.
Which chemical?
There was epinephrine.
Oh, epinephrine and norepinephrine. So if they're producing that, would our bodies react to it? Good question. I don't know the sensitivity. Like, what's the concentration you need of those chemicals? Does anyone know?
Very low.
I think, right? Kind of like the hormones, it's, you know, small doses. So I wouldn't doubt it, that there might be some kind of physiological response. I don't know what it is. Doesn't that? Isn't it? Yeah, I was gonna say, doesn't it jack you up? It's kind of a neural thing. Oh, that's a really good question. I'd have to look into that to see if they've done any dose dependent studies on that. Any other questions with quorum sensing? Yes.
They're all secondary metabolites.
Yeah. Yeah.
They don't use them. Besides,
Well, they, yeah, I think they might use them for signaling too.
so does that mean they're secondary metabolites still?
Yeah, so some secondary metabolites, it's a little gray. Sometimes they can be in the camp of a primary metabolite sometimes. It's a little gray sometimes with some of them, but most antibiotics have been considered secondary metabolites. Because they're not always being produced, which kind of makes sense. You wouldn't want to produce like streptomyces. One streptomyces species can make many different antibiotics. So if they had that up and running all the time, they'd be exhausted. You know, it would be just way too much. Because some of the physiological pathways to make these antibiotics are complicated, so. Okay, let's stop there for that, though. We'll get into the ecology of why they're doing this a little bit more after break, but I wanted to ask you a Google Forms. So just even if you have like a not much to say about the Google form, still answer it because this is participation points. Okay. So at least jot something down. This is to thank you for showing up today with break right around the corner. Last day of February. We're done with that. We're done. I'm not gonna do anything else though. Thank you for being here today. So I'm gonna talk to myself. And have a good break. That's what it looks like in your.